Welcome again, guys. Welcome again in another video of our Pedigree video lecture series. So from this video we are going to talk about some examples where you need to talk about whether this particular trait is autosomal or X-linked, or whether it is dominant or recessive. Okay. So today we are only going to talk about autosomal trait. That means our trait is a result of change in autosomal chromosomes. As I have told in our previous video, autosomal traits are result of defect or change in these 22 pairs of chromosomes, right? So first of all, you guys please check the rules that I have written here, and I am going to explain this via some example, because I think this could be much better rather than I will only talk you with I will only teach you these simple rules. So let's begin, guys. So this is our first example, and with the help of this example I am going to teach you how you can determine whether a pedigree is autosomal dominant or recessive. Okay, so this is a pedigree chart where this field symbol simply indicating us that they are diseased person, or you can say the trait they are phenotypically expressed by these individuals. And okay, so they want to know from us the mode of inheritance. So first of all, you need to distinguish whether it is dominant or recessive. Okay, this is your first job. For doing this, you need to know one thing: if there is a skip past of generation, you can conclude that it is recessive. What does that mean? That means it simply means that in some generation the disease will not be shown phenotypically, right? But so let's look here. So this is our generation 1, this is our generation 2, you can say this is our generation 3, and this one is our generation 4. Okay, so in generation 1 we have our disease person, in generation 2 we have our disease person, in generation 3 we have our disease person, and in generation 4 we have our disease person. So we can conclude that no matter it is autosomal or sex linked, but it is damn sure that it is dominant. If it is recessive, then we will have some generation where you will not find any field symbols. Okay, that means you will find something like that. But if there is a generation where you have at least one field symbol, then you can simply conclude it as dominant type. Then we will going to talk about whether it is autosomal or sex linked. But first thing is that to distinguish between whether it is a dominant or recessive, you need to find out whether the trait is showing skip past generation. Okay, that means in some generations there will be in some generations of the family the trait will not be expressed, and that is the reason why one of the main rule that I have written in the previous page, that is in case of autosomal dominant, affected parents can have unaffected children, right? What does that simply means? That simply means that if this affected suppose it is affected parents, but those children are unaffected, but in case of autosomal recessive you have unaffected parents, but these unaffected parents can have affected children. Why is so? Because I have told you that in case of recessive types there will be skip past generation. So it is obvious that in previous generation the trait is not shown itself, but in the next generation the trait expressed itself, and that is the reason autosomal recessive type have some things like that. So first of all, you need to focus on whether it is dominant or recessive, and how you can distinguish. You can simply look for skip past generation. If the trait is showing skip past generation, then you can easily conclude it that the trait is a recessive the trait is recessive type. But if it is not showing skip past generation, then you can easily conclude it that it is dominant type. So here we can conclude that whether no matter it is autosomal or X-linked, but first thing it is dominant. Okay, so after confirming this, now we need to talk about whether it is autosomal or recessive, autosomal or sex linked, sorry, whether it is autosomal or sex linked, or more specifically X linked. Okay, for knowing this we need to know the one simple formula. So what it is: if the trait is equally distributed among sexes, let me repeat it again, if the if the trait is equally distributed among sexes, then we can easily conclude that the trait is autosomal. So let's see here. Here is a man affected, here is a man affected, here is a man affected, here is a man affected. That means 3, 4. So we have 4 mans affected, right? So here is a female affected, here is a female affected, here is a female affected. So we have 3 female affected. That means we can conclude more or less that the trait is equally distributed among sexes, right? But in case of X linked, you have you have something like that, that all men are affected, or you can say there is a greater proportion of men affected, not very small amount but significantly. There is a significant increase of diseased man rather than diseased female. If you have something like that, then you can easily conclude that this particular pedigree is an example of sex linked pedigree. But if you have find something like that, that where the trait is equally distributed among sexes, then you can easily conclude that it is an example of autosomal trait. So what we can conclude, that it is an example of autosomal dominant. Why dominant? Because there is no skip past generation. Why autosomal? Because the trait is equally distributed among sexes. So I hope it is clear. Then let's move to the next question. The next question simply wants to tell us that the state the genotype of individual 1 to 5. Okay, so let's talk about that. So as I have previously mentioned in the rules, we need to solve this answer with the help of genotypic analysis. So in case of autosomal dominant types, we have genotypic analysis something like that. Okay, so in case of homozygous dominant, we have our diseased individual. With the help of heterozygous dominant, as we all know that expression of capital A marks the expression of small a. So we can also conclude that in case of heterozygous state, we will also have the diseased person. Only when there is homozygous recessive, we can conclude that the individual is normal. Okay, specifically in case of autosomal dominant. Okay, so in case of autosomal dominant, the heterozygote will also be a genotype of diseased individual, right? But we will not going to see this in case of our autosomal recessive. In that case, the heterozygote will be recessive. Okay, where heterozygous will be what? Heterozygous will be normal, but in that case it will be referred to as carriers, right? So you can also conclude whether a particular pedigree is dominant or recessive by simply observing is there are any carrier or not. If there are any carrier in pedigree, you can simply concluded blindfolded that the pedigree is recessive. Okay, because in case of dominant pedigree, there is no existence of carriers. In that case, the heterozygous state is also a diseased person. So that is the simple idea for doing genotypic analysis. Okay, so in case of normal individuals, in the whole pedigree, as we have already concluded that the trait is autosomal dominant, we can easily wrote that they are small a 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 small a. Let me write those. Okay, so all the unfilled symbol have genotype small a, as we are talking about here autosomal dominant, and in this case both capsa and capsa small a showing or indicating the disease type. Only small a wants to tell us that they are undiseased, or we can say normal. Now again come back. So in case of field symbol, we can easily conclude that they have either genotype capse or capse small a. Okay, now by doing simply Punnett square, we need to conclude what we have to take, whether capse or capse small a. Okay, so let's do this. By simply observing with the help of observation power, we can easily conclude that as they have 3 diseased children and 3 normal ones, so we can easily take this one. No, sorry. If we can take this one, then Punnett square will give us something like that. A. So all the 4 individuals will be diseased. So we can not take capse. We have to take capse small a because they have undiseased children. So there will be genotypes like small a. Okay. But if we use capse, then there will be no probability for small a. So no individual will be normal, but they have normal individuals that simply tell us that this is the genotype for the diseased individual, or we can say diseased male is capse small a. Okay. Now come here. So we have already concluded that number 1 individual's genotype is small a. Now we need to talk about the genotype for 2 individual, and we have already concluded that number 3 individuals genotype is small a. So we need to do this. So let's start. Now watch that thing, that there is also a confusion here. If we take capse, then all individuals will be affected. Why? Because if we do Punnett square, then we have all progenies heterozygous, and as I have previously told you that if in case of autosomal dominant we have heterozygotes, then we can all the 4 progeny will be diseased, but they have normal progenies, right? That simply wants to tell us that here also the genotype for individual 2 is capse small a, so that when if this capse small a individual mated with small a female, then they have 2 normal child. Okay, so we have concluded that. Now there is a question that what will be the genotype for individual 4. Now let's look here. As we have our genotype for individual 2 is capse small a and individual 3 small a, by doing Punnett square there is no probability that we have something capse, because for doing this female need to have something some capse, but she doesn't. That means by doing Punnett square we will not have something like capse, we will have capse small a, or we will have small a. So the genotype for individual 4 will also be capse small a. So your answer will be something like that: small a, capse small a. The last question is, if individual 2 and 3 have another son, then what are the chances that this son will be affected? 50%. Capse small a. By as they have simply given this, so we can conclude that our answer is right, because we have also derived that the individual 2 genotype is capse small a and individual 3 genotype is small a. So let's talk about this. If they have another children, then what will be the possible chance that another children will be diseased or not? Let's talk about this. So for this we need to Punnett square this. Okay, so a small a capse small a. Now look at this. As we have our capse small a, we have our small a, we have our small a. So all these 4 probabilities is completed, right? Okay, so there is a high chance that the next one will be capse small a. Okay, so we can conclude that the chance for the number 4 mane or we can say that as they have mentioned number 5, then we can mention number 6. If there is a progeny number 6, then by simply doing Punnett square we can conclude that the chance of the next progeny will be affected is simply 2 by 4. Now you will say why? Because simply let's look. They have capsa small a 2 and small a 2. So the total number of progenies is 4 and the number of disease progeny is 2. So 2 by 4 is equal to half. That means 50% chance that the next progeny will be affected if they have a children again. Okay. So this is an example for autosomal dominant. In our next video, I will going to discuss with you example for autosomal recessive. I hope it is helpful to you and you have well learned how to do genotypic analysis. If it is helpful, please like this video, share with your friends and subscribe my channel. And if you have any doubts regarding genotypic analysis, because I know it is little bit difficult, I will also talk about this in our upcoming several videos. So you don't need to worry, and if you have some doubts you can comment this in your comment box. Okay, so bye bye, take care.